today's presentation. Yukie Akune is going to talk about CarbArrayART, a software she has developed over the last years and which can be used for the interpretation for the organization and interpretation and analysis of Glycan array data. Yukie is working at the Imperial College in London at Jan Liu and Tenfesi's group. And with that, I would say, Yukie, please take it away.
Thank you, Renee, and thank you, Aku, for organizing the webinar series. Excuse me. Okay, so yes, today is my second talk in the entire series. Thank you very much. Let me go to okay. So yes, my talk today's and thank you very much everyone for joining today's webinar. I'd like to introduce the usage of CarbArrayART, the software for day-to-day data storage and data presentation, also data sharing software tool. Before going to the software, I'd like to start with the background of our laboratory, also CarbArrayART. In our lab, we have over 900 glycan probes, which are lipid-linked glycolipids and neoglycolipids. And this is kind of basic categories we have on our libraries. Majority are mammalian type, and some of them are like a fungal or bacterial plant-based polysaccharides. And how do we use them is, these are the example of pre-designed glycan microarray slides, but we print our library 900 glycan probes to create the big screening arrays. Oh, sorry. So this is the illustration of our slide layout, and each color indicates different types of subarrays, which I'm going to describe a little bit more in the later slides. But we print maximum 64 glycan probes per block, per square. So this big screening array has about 900 glycan probes. We also have several types of focused arrays for the specific glycan binding samples, like glucan, oligosaccharides focused arrays, or N-glycans focused arrays or mucins, etc. So this, to handle those data resources from the earliest stages, the in-house microarray software tools are required to manage them, to store the information, and to interpret the glycan microarray results. And the software tools were developed by our colleague Mark Stoll, and they are the mainstay of data management and reporting in our lab. This is an example of data processing using Mark Stoll. So Mark Stoll's software. After the experiments, the data will be saved in the software to generate charts or tables or heat maps. So this is the basic idea of using software to manage the data for the data presentation in your computer. However, the Mac software is very powerful, very useful software, but it has been developed in stages using Microsoft Office and Visual Basic, and they were not readily distributable, and there was some limitation on its extensibility. So we developed robust and distributable glycan microarray software, CarbArrayART. The CarbArrayART is a plugin of Grids Toolbox, which the collaboration with Renee and Senna, we have developed the CarbArrayART. This is very powerful method, algorithm with Grids. Sorry, the Grids is the software tool for mass spec data. and which have very strong data management tools. So CarbArrayART also can use the some of the functions like project management, sample management, experiment management, but we implement them for and we developed a new scheme for Glycan microarray data. Okay, so this is the so far it's the background. Sorry, and I'd like to move on to the software usage part. This is a web page where you can download the software, but also you can also have a look for the manual, user's manual or support information. Because there's a limitation on the time, I cannot explain everything today. So if you are interested, please have a look in the web page, and yeah, you can download and try the software. So before the software usage, I'd like to share a kind of minimum information you need to have in your hand before to display the Glycan microarray results in CarbArrayART. The first one is Glycan probe library, the Glycan probes which are printed on your microarrays. And then Subarray layout, the arrangement of the spot locations on one subarray. And then slide layout, the arrangement or designing of the subarrays in one slide. Maybe it can be one single huge pad or one huge subarray in one slide. It can be multiple subarrays in one slide, like 2x8, 4x8, 4, or anything's fine. Also, you need to create the project. So this is a kind of data folder name. It can be anything. We write here as a project, but it can be collaborator's name, the date of the project, date of the, anything, the experimental's name or anything is fine. Analyte, in CarbArrayART, analyte stands for the Glycan binding samples. So the analyte information, like your Glycan binding sample name, or sometime protein name with batch information, or protein name with the date or anything. Then the last information you need to have is Glycan array data, the quantified array data you generate from your scanner. And it can be GenePix scanner or ProScan scanner, it's fine. This is the data management workflow of the microarray analysis. The left side in yellow background is the workflow in CarbArrayART, and the right side is the workflow in array in experiment. CarbArrayART is MIRAGE compliant software, data storage. For the MIRAGE document, it was introduced in the last talk, so I'm not going to talk any details, but please watch the video or please go to the paper if you are interested. Then CarbArrayART is for day-to-day data storage, also data presentation in tabulation charts or heat maps. Also data sharing through the other CarbArrayART software, or exporting as PDF, Excel file, or Word file. So here, yes, I'd like to run through the 7 tools, but again, there's more functions, and I'm not sure how much I can explain in this next half an hour. So for more information, please have a look on the user's manual. And then I'd like to start with Glycan probe entry tool. So the Glycan probe entry tool in the CarbArrayART, it has 3 sections. One is Glycan probe entry, and tag section entry, and Glycan section for the Glycan moiety entry, which I'm going to show in the next slide. In CarbArrayART, the item with asterisk indicates the mandatory information. So in this example, in this entry, Glycan probe entry tool, only the Glycan probe name is mandatory information. And also the text in blue font, which are the items compliant with MIRAGE guidelines. So which means this information is highly recommended to record in your CarbArrayART software. Okay, so in the Glycan probe entry section, you have to enter the Glycan probe name, but there's also as option, you can put the backbone type information. This list has been arbitrarily made in our laboratory. This is very useful if using this category, we can categorize the Glycan probes without sequence information in a tabulation view or chart view later on. So, for example, if you have a Glycan probe, but the sequence is not fully defined, but if you know the Glycan is originally from, the Glycan is GAG-related Glycan, you can still categorize them as, okay, this is GAG-related. And later, you can filter or sort the results, the table, according to this backbone type. And the tag section is name and the text sequence information in text. You can also record the tag nature as a natural, synthesized, or unknown. Then moving forward to Glycan section. If you know the Glycan sequence partially, fully, you can enter the Glycan sequence information using this text sequence format. If you enter the sequence, you can search the glytoken ID. Also, if you have the glytoken ID, you can enter the sequence using the glytoken ID. If you don't have glytoken ID, once you enter the sequence and search the glytoken ID, and if it's not matched with the glytoken database, of course, nothing will be returned, which means you have the chance to register your sequence into the glytoken database. Okay, so this is the way, how to store the Glycan probe information in CarbArrayART, and you can create the Glycan probe library in your software, in your CarbArrayART. Then after making the library, you can create the Subarray layout. This is an example. This is an example of the Subarray layout. It has 28 Glycan probes in one subarray with 4 levels, 0.3 femtomole per spot represented in green, 0.8 in orange, 1.7 in blue, 5 femtomole per spot in the brown. And each Glycan probes are arrayed twice, so they are duplicate spots. And this subarray has 16 by 16 spots. This is our laboratory's default arrangement. So in this case, we printed 28 Glycan probes, and the last 2 rows are just empty. This is an example of array. Then this is screenshot of the entry page 1 of the Subarray layout tool. So the name, mandatory information, is ganglious dose response set, and number of replicates is 2. Number of labels in the subarray is 4. Number of Glycan probes printed is 28, and the number of spots in the subarray is 16 by 16. Then moving to the page 2, which has 3 tables. The top one is to define the concentration or dose of Glycan probes arrayed. You can select the unit of labels by the list, by the pull-down menu listed here. Then the table below is the list of Glycan probes which pre-stored using Glycan probe entry tool. Then the last table is to define the arrangement of Glycan probes arrayed in your subarray. So the first 2 columns are the row number and the column numbers of the subarray. The number indicates the spot starts from the left top corner to the right bottom corner. So 1 to 1 means the corner of the top left. The one, row 1, column 2 is the next one, going to the end. Then Glycan probe column is the Glycan probes arrayed, and the label, the number 1 to 4 is indicated here on the top table. And the last column, identification number of Glycan probes in the subarray. This is we call group number, which is the group number is used when the CarbArrayART calculates average intensity values in the tabulation result view. So in CarbArrayART, how we calculate average intensity values is if the Glycan probe IDs are the same and the group number, this identification number is the same, and if it has the same level, same concentrations, then we calculate the average intensity values. So in this example, GM4 has 4 levels. So it has, it will, in the tabulation result view, it will have 4 different intensity values according to their levels. So this group number, identification number, is really useful if you have, sorry, this is animation to how to make it. Yeah, if you have same Glycan probes printed in one subarray. So maybe first one, GM4 printed here, but maybe in one case, one example, maybe we need to reprint the same GM4 to the quality assurance or for test print or something in one subarray. But you don't want to mix the average intensity values of 2 of them. So in this case, using this group number, you can distinguish them. So this is a way of how to design the subarray layout in CarbArrayART. Moving forward to slide layout. Then slide layout is the arrangement of the subarrays in one slide. So in this example, the name, ganglious dose response set, and then the column and rows of subarrays used in one slide is such 2 by 8. And in this example, we use the same subarray layout to all the subarrays positioned here. So it just copy to all the locations. That's it. So this is the end of the slide layout tool. This is simple. Okay, so now I quickly show the 3 entry tools, but you can also use the Galo-like file. If you're familiar with GenPix scanner, maybe you use the Galo file, which contains the block number, spot numbers, and Glycan probe ID and names. You can add a little bit more information, the concentration or dose of the arrays printed, and you can use it into CarbArrayART to create your own library. Okay, and I'm sorry, moving forward because yeah. So this is the end of the library creation, that how to make the Glycan probe library, slide layout, and slide layout. Then I'd like to move to the sample metadata. This is more like lab note. You can record the sample information, sample metadata in CarbArrayART. Just for your lab record, but also you can use for data presentation or data reporting. So here, the metadata entry tool, this is a page 1. You can create from scratch, or you can copy. If you have entered previously, you can copy from your history, but also you can use the pre-saved templates, which are based on the MIRAGE document we created. And it has basically 4 sections: the sample information, tracking information, purity or quality control information, and amount of the samples. And yes, then the item compliant with MIRAGE guidelines as labeled at MIRAGE in the guidelines column. And here the items highlighted in red is the unique items, so you can enter only once per Glycan binding sample. This is an example of the entry. So this is, so you can select the sample type or species, or you can record a database entry or tag information, anything. Okay, now I'd like to move to the experiment protocol. This is, you can record the array Glycan microarray protocols in CarbArrayART. And this entry is created per Glycan binding sample. So 1 experiment per sample. You cannot create it, multiple experiments. Yeah, but anyway. So you can again create the experiment protocol from scratch, or if you have entered before, you can copy from your history, but also you can create the experiment protocol using the templates we saved as default in CarbArrayART. Yes, so this is the list of the templates. One is fluorescence labeled sample, biotin labeled samples, sequence detection, and tagged samples. You can start from here. You can modify the protocols. You can, just use the, if the procedure is exactly the same, of course, you don't need to change. But then after selecting the templates in this example, it comes to the designing tool page. It has the canvas area. So this is the protocol created in here. And then, okay, sorry. Yes, sorry. Yes, then The middle part is palette area. And in this area, each protocol are listed. So you can drag and drop to the canvas area to create or to modify your current procedures, protocols. And then, okay. Then this section is a parameter area. So this area is used to record the parameters used in each procedure, in each protocol. For example, in overlay protocol, the list of the parameters are here, and you can record the details information, the parameters of the experiment in CarbArrayART, such as what is the sample concentration or diluent composition or a temperature or time, volume, etc. Here the same, the items compliant with MIRAGE guidelines are highlighted, labeled as MIRAGE in the guidelines column. So this is a experiment protocol design. And yeah, I'm just going to repeat that. You can create your own protocol, of course, and you can share the protocol with your colleague, or you can include in your report as well. Okay. Then I'd like to move on to the data presentation view. So after entering the library, after entering all the sample information or the experiment protocol, fine, you are ready to make the tabulation result view. How to create it? You need the scan result file. The files are generated after scanning the slides. And in CarbArrayART, the GenPix results 3, version 3, export version 3, and version 2 as well. And also Analyte It version 6 and ProScan results are used. We can use those files to create the tabulation result view. Okay, so this result view is created under Analyte Glycan binding sample information. One sample can have single or multiple results. So yeah, you can repeat, if you have experiments repeated many times, still you can save under the same Glycan binding sample. This is an example, a screenshot of the entry page 1 of the Glycan array data. You need to enter the name of the scan data. Then you need to select the file type, either GenPix or ProScan or Excel file. And as I shown in the earlier, the third page 3 or 4 of my presentation, one experiment can have multiple slides to test at once. So in this section, you need to identify how many slides you used in your experiment. I think majority 1, or can be up to any number. Then the statistical method. Yeah, it can be average or it can be elimination. Elimination means to exclude the highest, maximum, and minimum values removing the maximum and minimum values per Glycan probe. So if there is 6 replicates per Glycan probe, you can average after eliminating maximum and minimum 2 spots. Then you can calculate the CarbArrayART will calculate average intensity values after elimination. Then signal to use, you can select which signal you'd like to use for calculating the average intensity values, either median minus background, mean minus background, or median or without background, or mean without background. And then actually I skipped some, but after selecting the slide layout, also file uploading, you can you need to select the which fluorophore you used in the scan file, because in GenPix or ProScan as well, there's a several fluorophore can be recorded in one file. So you need to select which one you'd like to use. Then the tabulation result view is automatically generated based on the slide layout you selected and GenPix or scan file you uploaded. So on the top section, there's a parameter sections. Still, you can change. You want to how you want to display, how you want to calculate the average, the intensity values. Either average or elimination, you can change later. You can change the scan power, or you can change the fluorophore you used. Then, according to the parameter, the master table will be updated automatically. So the master table in the middle shows the glycan probe name, ID, the structure if recorded in a symbolic format if available, and then intensity values per glycan probe levels. Then this section is a filter and sorter sections. You can sort or filter the master table results according to the number of specific monosaccharides or backbone type, or the linkage information of the stearic acid, or the even motif. You can filter the results. Then there's also the tool buttons to create the histogram view at the bottom, but also you can export the table to the Excel file from here. Yes, this is a list of the glycan motifs and monosaccharides used in the CarbArrayART filter and sorter sections. There's 78 types of monosaccharides and 72 motifs, and you can also categorize. You can use as the acidity of the glycan or monosaccharides, and can be number of branch or number of monosaccharides, etc. You can also create the heat maps by using the comparing tool in the CarbArrayART. So in this example, the glycan probes are aligned according to the glycan binding samples tested. So you can make a huge table against the per glycan probe, including the different array results, to create the heat maps, which is in this example, the heat maps are generated in using the Excel file, but the Excel file is included in the CarbArrayART software packages, and you can use it. Then, yes, the last part is data sharing part. So in CarbArrayART, how we share the data is you can share the data by selecting the project. And if you are sharing the data with collaborators, for example, and you want to include everything, like sample information, protocol information, results, plus any archived data, or like image file or references, also any, like even the multiple samples tested, everything included one project, one folder, can be shared to the collaborators' CarbArrayART software. Or you can create the Word file, PDF file based on the tabulation results. So once you part experiment results, tabulation view, or process the data, you can create the Word file or PDF file, which can, this is option, which can include analyte information, Glycan binding sample metadata, and experiment design. Yeah. So this is an example, the screenshot of the exported Word file, which has the table of the sample metadata and experiment protocol, the workflow, then experiment protocol metadata within one file. Yeah, so this is almost there. So this is the kind of tutorial I want. Yeah, the 7 tools I listed here in the earlier page. But also there's more functions, and there's some more tutorial videos in the web page, and more videos that we're making right now. And yeah, I hope yeah, we will upload more videos sometime soon. Very briefly, the future directions. We are now working on the CarbArrayART version 2 to be able to handle the transport for the data, the to and from the repository, Glycan microarray repository, which will be introduced in the Renee's talk in next month, I think. Then I'd like to, yeah, for my last page, I'd like to acknowledge my colleagues. And yes, this work was supported by Wellcome Trust and NIH, also much of DIMES and MRC grant. And then thank you very much. And next, yes, this is my last slide, and I'd like to finish my talk here. And the next talk will be Akune,